Module 8, Gene-Diet Interaction. In this figure, you see that health status results from the complex interaction of many different factors that intersect and combine to determine health status. This puzzle analogy is really quite appropriate because our health status really is the result of these interlocking factors that work together to determine our health status not only at any one time, but throughout our lives. In answer to the question, genes or environment, a famous author once wrote, "Eminent physicians have decreed that energy is born and not made, that humans, through the secret formulas of heredity, receive an impetus at birth which sets the pace for their bumpy journey across the moral span. In a general way, then, the life force is influenced more by ancestors than by vitamins." The author was Robert Louis Taylor in his book on Winston Churchill, An Informal Study of Greatness. Sir Winston Churchill lived in a very violent and stressful time. He drank his favorite whiskey and soda every day, and he smoked Cuban cigars whenever possible. And he died in 1965, shortly after his 90th birthday. Jim Fixx, on the other hand, almost single-handedly began the running craze of the 1970s and 80s that still goes on today. He transformed himself from an overweight, out-of-shape businessman to one of the world's most recognized fitness gurus. Yet he died while running at the age of 52. Nevertheless, his dad had died at the age of 42, and Fixx took up running fairly late in life. Despite his early death, his change of lifestyle was probably a key factor in extending his life 10 years beyond his father's. The critical factor in both of these instances was most likely the contribution of genes. In the case of Winston Churchill, clearly his family is segregating genes for resilience and longevity, and in the case of Jim Fixx, he actually had a family history of heart disease, and he had lived most of his life as an overweight, out-of-shape businessman. The fact that he took up running changed his environment, but his genes still had a strong role to play in his health outcome. So genes work together with all of these factors to determine our health, and that's the foundation of gene-environment interaction. As we've discussed earlier, many of our traits are highly heritable, the most heritable of which is probably height, but weight and systolic blood pressure and many of the traits that we measure to determine our health status are also very, very heritable, including longevity itself. In our studies of genetic analysis, we've seen that the more complex the trait and the more normal the distribution of the biologic trait, the more likely that that trait is encoded by more than one gene. There are many types of gene interactions. Pleiotropy refers to the fact that genes can contribute to the outcome of several traits. And epistasis refers to gene-gene interaction, instances in which multiple genes are interacting to affect a trait, and that multiple traits are interacting to produce fitness in the organism. Therefore, natural selection leads to gene combinations that determine multiple traits. The two-hit model of cancer is actually a great example of gene-environment interaction. In this model, the cells are protected by two copies of the genes that ensure the reliability of cellular replication. When one of those genes is knocked out, the cells become susceptible to cancerous growth, but not yet manifesting that cancer. A second hit occurs when the cells are exposed to environmental toxins or radiation. And when that second hit occurs, that's when the cell no longer has its guardians and cancerous growth can occur. So what defines an environmental or a genetic disease? In the case of a purely environmental type of condition, that might occur if something like a meteor struck. That would clearly cause a lot of damage and disruption, but there would be nothing that your genes could have done to prevent you from being pretty significantly damaged by a meteor strike. On the other hand, sickle cell anemia is an example in which genes have everything to do with whether or not that defect is manifested. In a complex disease like obesity, for example, probably lies somewhere on the continuum between purely environmental and purely genetic. This figure outlines the contributions of genes and environments in terms of a threshold of disease. In the first bar, the disease is primarily genetic because all it takes is the effect of genes to exceed that threshold of disease. In the second bar, the gene is primarily environmental. Genes have very little to do with it, and genes by themselves could not exceed that bar, a threshold of disease. And it takes the additional contribution of environment to exceed the threshold. In the case of the third bar, the combination of genes and environments must occur for the threshold to be exceeded. And in the case of the fourth bar, neither genes nor environments are sufficient to cause disease. In this figure, genotype and risk factors are depicted as they determine disease outcomes. In genotype A, the genotype influences the risk factor primarily, which then has an effect on the disease. In genotype B, the risk factor primarily influences the disease, and the genotype modifies the effect of the risk factor. If for genotype C, the genotype primarily influences the disease, and the risk factor modifies the effect of genotype. In genotype D, genotype and risk factor combine to influence the disease outcome, and in genotype E, genotype and risk factor independently influence disease outcomes. In terms of nutrition, nutrition can act at the level of the risk factor and at the level of disease. We know that many genes and environments interact to influence our health. Certainly diet and exercise have a big part of our health. In terms of energy balance, we know that the weather, the physical environment, and global warming, which is keeping all of us indoors, may influence our physical activity behavior. And certainly environmental toxins and radiation have an influence on our health. And even drugs, which are meant to be curative, may often induce weight gain or other side effects that influence health as well. And certainly our metabolism and our microbiome in our gut are big factors that we know influence overall health. And all of these interact to paint the picture that will be our health within the context of our genes. As we saw earlier, interaction can be depicted by stratified types of analyses in which you see, in this case, genotype effects that are distinctly different given the environment in which they're examined. Interaction can also be depicted like this, with the classic lines that cross that in terms of regression represent interactive effects, in this case between diet A and diet B. Does genetic variation influence response to changes in the environment? This is a classic study of gene-environment interaction in which 12 monozygotic identical twin pairs were overfed 1,000 calories a day for 84 days. So these young adults were paid to sit around, they were asked not to exercise, and they were overfed. So it was every grad student's ideal dream study to be a part of. And what they found, without looking at any genes or DNA sequence, was that there were significant correlations between the twin pairs for changes in body weight, percent body fat, and fat mass, and that there was 6 times more variability among the pairs than between them. So high concordance between the pairs for weight gain and changes in percent fat and fat mass. In the second study on the right, they actually put the twins then in negative energy balance, not by making them diet, but by giving them an exercise protocol of vigorous cycling on a cycloergometer. And again, they see this very tight concordance between the twin pairs, actually even less variable for weight loss than it was for weight gain. As they put them in negative energy balance. And what they concluded from these studies is that genetic factors interact with these changes in the environment to influence how we respond to them. Genes and environments interact to influence nutrient homeostasis as well. As nutrient levels go up, they feed forward to the enzymes that utilize them in a feed-forward mechanism, thus ensuring that as you overeat, for example, lots of fat or carbs, the enzymes needed to metabolize those fats and carbs actually go up. And as you reach homeostasis with the new levels of the enzyme, then ultimately the levels of the nutrient in your system would be metabolized more efficiently and would go down. As nutrient levels fall, that would provide a feedback mechanism that would also decrease the enzymes that would utilize them, which would ultimately increase the levels of the nutrient in your body. So even metabolism is controlled through this combination of genes and environments changing and feeding backward and forward on one another. Nutrigenetics refers to the interaction between genes and diets. So in this example, we have a C to a T nucleotide sequence variant. And we have 3 individuals, a CC, a CT, and a TT. And prior to any dietary intervention, we see that they all look pretty similar. In the case of gene-diet interaction, we give them some kind of test meal. In this case, maybe a high-fat, high-carb kind of hamburger. And we see that the CTs and the TTs respond very differently to this diet, providing an example of gene-diet interaction. The ApoE gene actually provides a great example of gene-environment interaction. Variation in ApoE is associated with cholesterol metabolism, and it's also associated with altered risk for heart disease and for Alzheimer's disease. The classic genotypes of ApoE result from the combination of actually 2 SNPs. And here on the upper right is depicted these alleles that have been genotyped historically with kind of some very old school enzyme restriction assays, generated these combined alleles called epsilon 1, epsilon 2, epsilon 3, and epsilon 4, or E1, 2, 3, 4. And the ApoE is associated with the clearance of both cholesterol and of amyloid plaques from the circulation. The most frequent allele of the combined allele is the E3 allele. E2 is associated with increased activity of ApoE, and E4 is associated with decreased activity of ApoE. So the E4 allele puts you at risk for greater risk for heart disease as well as for Alzheimer's disease. So in this study, Boerwiegel and colleagues gave their subjects a postprandial test meal challenge. And this was a lovely drink that consisted of 350 grams of heavy cream, 2 tablespoons of chocolate syrup, a tablespoon of granulated sugar, and some instant non-fat milk, and a little bit of vitamin A to help metabolize all of that fat. And altogether, this very fatty test meal drink contained 1,374 calories and 130 grams of fat. So very, very high in fat. So they gave this test meal challenge and basically wanted to see whether or not subjects responded differently by genotype to this test meal challenge. When they looked at the African Americans to see how they responded to this test meal, you can see that there were no differences between the ApoE33, which is the wild type version of the genotypes, versus E34. Pretty much you see a rise in retinal palmitate with a little bit of leveling off, which is what you'd expect. But when they looked at the ApoE23 genotypes, they saw this tremendous rise that continued to rise throughout the course of the study. And when they looked at triglycerides, what they saw was that the ApoE33s saw a rise in triglycerides with a fall after about 4 hours, which is what you'd expect. In terms of the 2,3s, you see a rise and pretty similar to the 3,3s until 4 hours, but then they level off and they never start to fall. And in terms of the 3,4s, significant rise in triglycerides that continue to rise slightly up to 8 hours following the high-fat meal. So what they showed was that there was a significant interaction between genotype and response to this high-fat meal. Another example of gene-diet interaction is this classic study of the ApoA2 gene. And you may recall this gene because it was part of a diet report that you saw earlier in an earlier module. In this study, which is a really very nice study, they showed that ApoE genotype was very dependent upon its effects on outcomes. In this case, BMI, was very dependent upon the dietary intake of the individuals. In the top left-hand corner, you see the significant interaction between genotypes of two different kinds. The squares is the steeper line and the circles is the less steep line, but a very classic example of an interaction in which genotypes are responding very differently to saturated fatty intake over the course of a range of BMI. In panel B, they're showing again that BMI is very different by genotype, but only dependent upon whether or not you're eating high or low fat levels of saturated fat. So the two bars on the left are the low saturated fat intake, no significant difference, and it's only a genotype risk-raising effect for BMI in the two bars on the right, where you see this significant difference based on genotype in the groups that were eating a high intake of saturated fat. In the panel C, you basically see this rise in BMI associated across the Framingham outcomes. No significant differences by genotype, but when you divide them by the saturated fat intake in the panel D, what you see is that now you have this very significant effect of genotype with the CC genotype combined with the high saturated fat, giving you the highest BMI. And what's nice about this is that that effect is consistent over time. So it's just one locus, but they showed this in multiple populations, and that makes this study pretty important. In terms of exercise, genes may have different effects in exercising versus non-exercising individuals. And there can be a large variability in how individuals respond to exercise training. And this is actually some of the work that we do in my lab. So this is a classic study. It was published quite a while ago now, but it was one of the first to show this interaction between risk-raising alleles, at this case for the ADRB2 gene, and different measures of body size and adiposity. And basically what Muirhead and colleagues showed was that the risk-raising effect of this particular polymorphism, GLIN27 was only apparent in those people who were not physically active. So in each of these panels, the top left is for weight, the bottom left is for BMI, the top right is for hip, and the bottom right is waist to hip ratio. You only see this difference in genotype in the individuals who were not physically active. Similar to the effects of that's seen in the Muirhead study for the ADRB2 gene, these are data from the Helena study of the FTO effects. So FTO, as we've talked before, is very strongly and consistently associated with obesity across many populations. What these investigators shown is that the SNP that has been associated with BMI has its strongest effects in those individuals who are not physically active. So the top graph is for body mass index and the bottom graph is for body fat. And what you see is that the sets of bars on the left represent a risk-raising effect that is only observed in the inactive participants versus the sets of bars on the right are for the active participants. Very clear gene exercise interactions. In our own work looking at the FTO gene, we genotyped 13 variants across the gene for association with exercise dropout. And what you see outlined in yellow are 5 of the 13 variants that were associated with increased risk for dropping out from exercise. That in itself is interesting, but what we did then was we added up and created a genetic risk score for all 5 of those variants and showed that any 3 alleles in the combined genetic risk score raised the risk of dropout more than twice that of the reference group. So pretty significant effects. This is the SNP that was originally shown to be associated with obesity, and this is in fact a very, very big gene with a lot of distance between the first and the last marker here. So we're continuing to examine this, but clearly there's something interesting going on with FTO and exercise. Finally, we know that genes and diets interact directly through the action of nutrients binding to transcription factors and other regulatory elements of genes. We know that fatty acids, glucose, hormones, and micronutrients can serve as cofactors and modifiers of gene transcription, and that provides a very obvious and functional gene-environment interaction in influencing the expression and translation of genes themselves. And circling back to our original example we began with at the beginning of this lesson about Jim Fixx and his amazing running career that ended abruptly in a heart attack while he was running. He became everyone's favorite reason to not go and run. Why should I run? It's all in your genes anyway. Except as we said earlier, he probably extended his life way beyond his father's because he was so strong and healthy later in his life. And here's an example of the next generation, John Fixx, who carries some of that genetic susceptibility for heart disease. He's been running his entire life. He's been a track coach. He leads a private school, and he's been a happy and healthy and still running today person who will definitely have changed his health outcomes through a lifetime of fitness.